Hello and welcome back. In this segment, we will describe two interesting applications of NGS to medicine that go beyond the common applications of DNA and RNA sequencing, which we discussed in the previous segments. The first is NIPT, which stands for Non-Invasive Prenatal Testing. Before this technology, testing genetic material from the embryo required an invasive procedure such as amniocentesis or CVS. These invasive procedures come with a risk to the pregnancy. To understand the novel non-invasive technique, we should realize that human blood contains cell-free DNA, DNA fragments with lengths of roughly 200 nucleotides that are released from damaged cells. It was discovered that about 10% of the cell-free DNA in a pregnant mother's blood actually comes from the fetus. It didn't take long for a smart scientist, Yook-Ming Dennis Lu, to realize that combining this observation with NGS technology could enable non-invasive prenatal testing. The procedure starts with isolating and sequencing cell-free DNA from the mother's blood and aligning the reads to the reference human genome. Half of the DNA sequence of the fetus comes from the father, but the similarity between the genomes of all humans is such that the vast majority of reads will be mapped to the appropriate genetic locations, regardless whether they come from the mother or from the fetus. Now, let's see what would happen in the case of healthy fetus. Each chromosome will be mapped after normalization by the length of the chromosome, and roughly uniform coverage will be observed, with about 10% of the reads coming from the fetus. Note that we don't care which read comes from the mother and which from the embryo, making the analysis that much easier. Now, let's assume that the embryo has trisomy, such as Down syndrome, with 3 copies instead of 2 of chromosome 21. In this case, the coverage of chromosome 21 will be 5% deeper, as we will have 50% extra DNA from the 10% of the fetal-derived reads. It may be difficult to realize from the schematic figure, but due to the huge number of reads in the NGS procedure, a difference of 5% is very significant and can be detected reliably, with both specificity and sensitivity of more than 99% for Down syndrome. Thus, by only extracting peripheral blood with no risk to either the mother or the fetus and using a very simple computational procedure, we are able to detect trisomy or other chromosomal duplications or deletions. The method can be applied not only to identify additions or deletion of a complete chromosome, but also to detect copy number variations of part of chromosomes, which occur in quite a large number of genetic disorders. This method cannot replace direct sequencing of the baby's genome to detect, for example, monogenetic diseases. However, it is approved by the medical authorities in many countries as a screening tool for large-scale chromosomal abnormalities. As a result of the availability of this procedure, the number of invasive procedures have been significantly reduced. To my mind, this is an ingenious application of NGS technology. Finally, let's return to the human microbiome. Our body provides a habitat for a huge number of bacteria. We estimate that there are more than 1,000 species of bacteria living in humans. And while the number is still subject to debate, it is clear that we have more bacterial cells within our bodies than human cells. In recent years, many studies have started to explore the interplay between humans and their bacteria. Clearly, the first step is to identify all the different bacteria in our body, an ongoing effort that is known as the Human Microbiome Project. To this end, we need to take a sample from the gut, the vagina, the skin, the mouth, and sequence the bacteria we find. However, unlike the human genome, almost all of these bacterial species have not been characterized or sequenced, since many don't grow in our lab cultures. One approach would be to sequence each of these bacterium de novo, as we cannot align them to a reference genome. We will not go into the details of the algorithm, which is called shotgun sequencing, but it is based on concatenating reads based on the overlap between them. This is how the first human genome sequence was created, and this is how we routinely sequence new organisms today. However, the problem of sequencing all of the gut bacteria, for example, is much more complicated, since we cannot isolate the different species and sequence them individually. Rather, the sequenced fragments represent a mixture of all the bacterial genomes, and obtaining sequences of each organism requires solving a huge multidimensional puzzle by reconstructing many genomes simultaneously. This field is called metagenomics, and it is a huge computational challenge. Thus, most current microbiome studies take a different approach. Part of the ribosome are very similar, but not identical, between all bacteria. Therefore, it is possible to use universal PCR primers and to amplify these segments. Then, we can sequence them and characterize a sample containing a mixture of bacteria based on the relative composition of each different bacterial phyla within it. Recall that a phylum is a higher level definition of taxonomy. Thus, we can differentiate, for example, between the phyla of Firmicutes and Proteobacteria, but we cannot differentiate between the species of E. coli and Salmonella, which are both members of the Proteobacteria phylum. Once we attain the phylum composition of the different bacterial communities, usually represented in what are called OTU tables, we can measure the similarity between the communities in terms of the diversity of their bacterial content. In this way, we can compare between the microbiome of individuals, for example, to follow the change in the microbiome during development from babies to adulthood to aging, or study the effect of diet on the microbiome. Another example is comparing the bacterial communities within and between various organs or locations on the body. In the figure, each dot represents a community of bacteria taken from a specific body area in a single individual, as characterized by principal component analysis. We can see that each environment in the body harbors a different community of bacteria, and that, for example, gut bacteria of different individuals are more similar to each other than communities taken from two different organs of the same individual. Many interesting claims have been made recently about the effect of the microbiome on health and disease, from digestive system disease to cognitive disorders. So far, the only approved microbiome manipulation treatment by stool transplant is for Clostridium difficile colitis, a disease which is mainly caused by a side effect of antibiotic treatment. This is a serious disease for which no good treatment was available. It turns out that transplanting stool from a healthy donor restores the healthy gut microbiome and cures the disease with over 80% success rate. But many other clinical applications are investigated. For example, there was a small-scale study published in a prestigious medical journal that showed a difference in the microbiome between babies that were born vaginally and babies that were born via a C-section. And it was suggested that this difference can be eliminated by applying a swab with vaginal fluid to the baby immediately after delivery. Many doubts have been raised about the benefit of such treatment, and currently the American College of Obstetricians and Gynecologists does not approve this practice. Maya and Ronen, the field of microbiome analysis is still in its infancy. There are many technical and computational difficulties in performing these studies, and the resolution of our available techniques is usually not good enough to identify specific bacterial involvement in disease. However, as you have seen, the field of NGS is rapidly advancing, and I am sure that in the near future we will be able to better understand the interplay between the human body and the bacteria that it hosts, and use this information for diagnostic, prognostic, and therapeutic applications.